In Chapter 13, we'll explore membrane channels and pumps. So just an overall outline, we're going to talk about membrane transport, and within that, we're going to talk about active versus passive transport. Through our study of membranes, we're going to look at 3 classes of membrane proteins that are responsible for the permeability of membranes. And these are going to be pumps, carrier proteins, and channels. The carrier proteins will be the secondary transporters. So the carrier proteins and channels are going to function through passive transport, whereas the pumps are going to function through active. So active transport and passive. So first we're going to go through and define what passive and active transport refer to. So there are 3 different classes of membrane proteins: the pumps, the carrier proteins, and the channels. And the carrier proteins and channels, we said, are going to function through passive transport. Pumps, we said, would utilize active transport. So what determines how a molecule moves across a membrane? Well, that movement will be dependent upon size and charge or polarity of the molecule that's trying to pass. So size and charge are going to be big. Now, passive transport will include both passive and facilitated diffusion. And with the passive diffusion, this we're looking at nonpolar molecules. So nonpolar molecules will move through passive diffusion, and this doesn't require ATP or membrane proteins. The molecules will simply pass through the membrane from high to low concentration, or down the gradient. So that's passive diffusion. When we're talking about facilitated diffusion, we also don't need ATP for this, but here we need a protein that's present to aid in the transport. So the molecules will move down a gradient, but they require a protein to facilitate the process. So overall, passive transport, molecules move down a gradient and do not require energy. So no energy is required when we have passive transport. They're moving down their gradient. When we're talking about active transport, the molecules will move against their concentration gradient, and they require both ATP and membrane proteins. So active needs energy, they require energy, and these membrane proteins. And usually this energy is in the form of ATP. Now the free energy, it can be stored in concentration gradients, and it can be quantified. So these graphs here are showing that it's actually the sum of information that determines whether or not molecules will pass. So what we're looking at in terms of the sum of information, we're looking at concentration ratios or concentration gradients and membrane potentials, so kind of charge across the membrane. And the gradient that forms will allow spontaneous movement of molecules through passive transport. In active transport, we're going to require energy because we're moving against that concentration gradient. And we do have an equation which will allow us to look at the free energy by combining the different terms that are associated with concentration and membrane potential. And here's our equation. RT ln of C2 over C1 plus ZF delta V. So here, we're looking at the free energy, so that's going to allow us to tell us whether it's spontaneous movement or if it's non-spontaneous or against the gradient. R is our gas constant. T is going to be the temperature in Kelvin. And then this ln, we're taking the ln of a concentration ratio, so we're looking at concentration on either side of that membrane. The Z term, this is the electrical charge of the transported species. So that might be a new term for you, but the others you should have seen before. And then we have F, which is Faraday's constant, and this value is 96.5 kilojoules per volt mole of electrons. And you should have seen this. We talk about Faraday's constant in general chemistry. And then we have delta V, which is the change in potential that's measured in volts. So we won't do much in terms of actually calculating the delta G here, but you should know that we can determine whether or not movement will occur spontaneously by finding the delta G, if we know the sign of delta G. and that all of these terms are combined together because it's a combination of concentration and membrane potential that's going to allow us to determine free energy and transport. So when we move large substances, we need energy to move these things against their concentration gradient. And this is typically done through the use of membrane pumps. The membrane pumps, remember, are going to use active transport because we need energy to get the process to occur. So the energy that's allowing us to move these things through active transport, usually we're talking about ATP, which is going to serve as the energy. Sometimes it's possible, more common it's ATP, but sometimes it can actually be light energy that's employed by these pumps for active transport. And the energy is required because we're moving against the spontaneous direction, or we're moving against the gradient. We're going to go through and talk about two different families of membrane pumps: the P-type ATPases and ABC transporters. Both of these are going to use ATP to move substances across the membrane. So let's start with the P-type ATPases. Now, P-type ATPases will couple phosphorylation with a conformational change. And that is going to allow that pump to function in order to move substances across the membrane. So here we're looking at this energy input here is going to be in the form of ATP. And we're looking at two different conformations. This is our membrane pump, and this is the substance that we're trying to move across the membrane. So here the pump is going to take up ATP from inside the cell. Okay, so this is the inside of the cell where our energy input is going to occur, and here is the outside of the cell. So in this particular case, we're looking at the pump getting energy from the inside of the cell in the form of ATP. That ATP is going to be hydrolyzed. So we're going to end up breaking that ATP down into ADP plus the inorganic phosphate. That is going to cause a conformational change in the pump. And that conformational change will help move the substance of interest, an ion in this case, across the membrane. So here the energy is transformed from energy that's stored in the bonds of ATP to energy that's associated with an electrochemical gradient because we're moving ions. The gradient that's created can also then aid in movement of other substances across the membrane through other means, which we'll talk about as we move through this chapter. So again, here substance, we have our membrane protein. Initially, it's going to be open to the inside of the cell. That's where our opening is. Remember, this is the inside of the cell. We have some small molecule that can enter in here. It enters into the protein. Usually it's going to bind or associate with that protein. Then we have the energy input, which is usually ATP. The ATP would be hydrolyzed. That's going to create that conformational change that takes this from being open to the inside of the cell to being open to the outside of the cell. And when that happens, the molecule that was in this little pore is going to be released to the outside of the cell. And it's ATP that drives this overall process. So one example of a P-type ATPase is this calcium pump. This is going to take calcium from muscle cytoplasm into the sarcoplasmic reticulum. And this particular pump has a single polypeptide chain that consists of our transmembrane domain. So here's our transmembrane domain. This has 10 alpha helices and a calcium binding site within this domain. Then we also see that the pump has three other domains: an A domain, a P domain, and an N domain. The N domain is where our ATP is going to bind. Because remember, these pumps, we have to have ATP coming in to allow this to function. And then the ATP is going to be hydrolyzed to produce ADP and that inorganic phosphate. So the P domain is what accepts that phosphoryl group when the ATP is hydrolyzed. The A domain that's shown here This is going to be a linker. It's going to link the membrane portion, that transmembrane portion, to the P and N domains. So this is just showing the same pump in a different conformation. And it's the conformational change that allows the substance to move across the membrane. Remember that the ion of interest is going to come into the transmembrane portion. We're going to have ATP hydrolyzed, and that's going to cause conformational change. Well, in this particular case, that conformational change will disrupt these calcium binding sites within this transmembrane portion, and that causes the release of calcium into the cell. This is just reiterating that overall mechanism. This is specific for that calcium pump. So here we have binding of cytoplasmic calcium. So from inside the cell, the calcium comes in. Then ATP is going to come in and bind to the N domain. ATP is then going to be hydrolyzed. And in that process, we're going to transfer that phosphoryl group, and it's going to transfer to a particular aspartate residue that lies within that P domain. So we transfer that phosphoryl group to the P domain. We have ADP being released. When that happens, there's a structural change that occurs. So we actually change that conformation of the protein. So the calcium binding site is now going to face the other side of the cell. We're on the outside of the cell. That phosphoryl group is then going to it's going to leave that P domain. Okay, we're going to get rid of that. And when that happens, the protein will change conformation so that it reverts back to the original position and can go on and bring more calcium in. This is just pictorially looking at what we just talked about. So protein starts in the unphosphorylated state. We call that the E1 state. We're going to start in this position. Here's our transmembrane portion, and then our A, N, and P domains there. We have our binding site in the middle here. So two calcium ions are going to come in, and they're going to bind that transmembrane portion. So calcium is going to bind, and it's going to bind on that cytoplasmic side, so the inside of that membrane. When that happens, we're still in the E1 state. We're still in that conformation. ATP then comes in and it binds to the N domain. So here's the ATP coming in and binding to that N domain. That ATP is going to be hydrolyzed. Okay. So we have that being converted to ADP and inorganic phosphate. And remember, that phosphoryl group is going to be transferred to that P domain. After this happens, the ADP is going to be released. The ADP is released, and that's going to also result in the, we have a conformation change that occurs here, and that results in the release of the calcium. And at this point, we kind of change conformation. Again, that conformation change, we're in the E2 state here. So once we're in the E2 state, we release the ADP and we release the calcium. The last thing we need to do is release that inorganic phosphate. Once that's released, we can then, that E2 state can revert back to the E1 state, and we can start the process all over again. This is similar to the mechanism that's employed by the sodium-potassium pump. This is just showing a particular molecule that's a steroid derivative that comes from plants. This is actually, this digitoxagenin is an inhibitor of the sodium-potassium pump. What it does is it will inhibit the dephosphorylation at that E2 state. So it prevents it from reverting back to the initial state. So this is referred to as a cardiotonic steroid inhibitor. And it can be used, this is just to illustrate an application of something that will impact these pumps. So this is used to treat heart failure because what it does is it allows for a decrease in sodium. We're not pumping the ions like they're supposed to, we're inhibiting the pump. And a decrease in sodium gradient will result in slower removal of calcium from the cell. because we'll see that all of these pumps are interrelated. If we end up with an increase in calcium, it's not removed from the cell as quickly, what that does is it enhances contractibility of muscle. Okay, so if we're looking at heart muscle, we're going to have better contraction of heart muscle here. P-type ATPases will play a wide variety of roles. There are actually 16 proteins that exist in the yeast genome that serve or are classified as P-type ATPases. In the human genome, there are 70 different P-type ATPases. All of these things will utilize ATP hydrolysis to move substances across the membrane. And all of these have to have some they're connected in some way, shape, or form because we need to balance out overall the substances on either side of the membrane. And the other type of pump we said we were going to talk about were the ABC transporters, the second type of transport protein pump. These are also called ATP binding cassette transporters. This type of pump, this class, also involves hydrolysis of ATP and conformation change, but it's going to have a slightly different structure and function in a slightly different way. So here we have 4 domains that arise from a single polypeptide chain. We have 2 that are going to be transmembrane domains, and we have 2 that are going to be our ATP binding cassettes that are inside the cell. So ATP binding cassettes. Both eukaryotic and prokaryotic cells will have these types of pumps. They may differ in the number of domains present, and that just depends on the type of cell that we have, but generally this is the structure. When we're talking about the eukaryotic cells, the eukaryotic cells will, with these types of pumps, tend to transport from inside the cell to outside. Whereas in prokaryotic, we tend to be transporting from outside to inside. Something like E. coli has 79 different ABC transporters. In the human genome, there have been found to be 150 of these types of substances. So if we're looking at the overall structure, the pump is going to begin in an unbound state, and that state is open to the cytoplasmic side, so cell interior. Here's our interior, here's our exterior. So the binding region faces inward. Substrate is going to enter the binding, it's going to enter in that binding pocket, and the binding of that substrate will induce a conformational change in the ATP binding cassette portion. So substance comes in, it induces that conformational change, so it looks different. Here's our open form versus our closed form, different conformations. That's going to increase the affinity for ATP. Okay? So 2 ATP can then come in and bind, and the binding of the ATP causes an additional conformation change that occurs in that transmembrane portion, and that will allow the substrate to be released to the opposite side. Once that happens, then we have subsequent release of that ADP and the inorganic phosphate, and that resets back to this form. So similar to what we talked about with the other pumps. In terms of the mechanism, we actually have 5 different steps. Only 3 are on this slide. First, the channel is open to that cytoplasmic side. Substrate binds, causes the conformation change in the ABC binding domain, or the ABC domain. ATP binds to the domains, and that causes additional structural changes. That orients the substrate so it's facing the outside of the cell. Then the outward facing conformation has a reduced affinity for substrate. So it allows the substrate to be released on the other side of the membrane. Then the ATP hydrolysis occurs. We release that ADP and inorganic phosphate that's in there, and it resets our transporter to the original state so that it can pick up more. This is showing pictorially what's going on there. It illustrates that mechanism we just outlined. So we have the cell in the unbound state open to the cell interior. Our substrate comes in and binds into that transmembrane portion. Then we have the binding after that of our ATP. That causes the conformational change. This causes release of substrate. The ADP is then going to be, so we have our release of substrate here. We're going to hydrolyze this, and our inorganic phosphate and our ADP will be released. That's going to reset the protein overall, and it can take more substrate in. I'm going to skip this slide for a second and come back to lactose permease. This is just slightly out of order. But we've talked about pumps that utilize ATP. Now we're going to switch gears and talk about secondary transporters. So these are all secondary transporters. These substances do not hydrolyze ATP directly. Instead, what they're going to do is they're going to couple non-spontaneous flow of one molecule to spontaneous flow of another. And there are three types of secondary transporters which are shown here. These are all collectively referred to as carriers. So the first one is the antiporter. The antiporter will use electrochemical gradient of one molecule to move a second type of molecule in the opposite direction. So for example, if A moves down a gradient, so A is moving down its concentration gradient, that will allow B to move against its concentration gradient. So the electrochemical gradient of one molecule is used to move another molecule in the opposite direction. One goes down its gradient, one goes against its gradient. The second type here is a symporter, which is also called a cotransporter. This uses spontaneous flow of one molecule down its gradient to move another against its gradient in the same direction. So if A is going down its gradient, the electrochemical potential that's driven is used to have B go in the same direction. The difference is this direction, B is going against its gradient. So A would be down its gradient, B would be against its gradient, but that movement occurs in the same direction. And then the uniporter, which is shown here, this is the same substance being moved in opposite directions. So in one direction it's down its gradient, in the other direction it's against its gradient. So these are the three different types of secondary transporters. So the lactate permease is an example of a symporter, and it's something that's found in prokaryotic cells. More specifically, we can see it in E. coli. The structure of this lactose permease, or lactate permease, it consists of two 6-membrane spanning alpha helices. In this system, E. coli will break down fuel and produce protons. It's going to result in high extracellular proton concentration and low intracellular proton concentration. So we're creating a proton gradient. The protons move down their gradient, they release energy, which is then used to transport, in this case, lactose, sugar. So the lactose can move from the outside to the inside of the cell against the concentration gradient. So this is just looking at the steps involved in that transport cycle. And there are 6 total steps. Again, some of them got cut off from this slide. But the first step is that the cycle begins with lactose binding pocket facing the outside of the cell. A proton binds. That's followed by lactose binding. Then the permease everts, or it changes conformation. If it was open to the outside, it's now open to the inside, or vice versa. Once that happens, the lactose leaves the permease and enters the cell. The proton then leaves the permease. That's step 5, the proton leaves the permease. And then in step 6, the permease everts back to complete the cycle. So here we're just looking at the structure of that lactose permease, the general structure. And we have that binding site in the middle there. And here we see that overall mechanism. We're starting with step 1 here in that mechanism. So the lactose binding pocket, that's going to face this is the outside of the cell. and this would be inside. So the binding pocket is facing the outside of the cell. We have this proton that's going to come in and bind. After that proton comes in and binds, then our lactose is going to come in and bind. That was step 2. Once that lactose is bound, we have eversion. So we're changing that conformation, and we're now open to the inside of the cell. Once that happens, our lactose is released. So we see that released into the cell. After the lactose is released, our proton is released. This should be empty right here. Once that proton and lactose are released, then we have eversion, and we revert back to the original form and can start the process all over again. Okay, so we've talked about pumps and carriers at this point. And these substances can move things across the membrane at rates of several thousand ions per second. Channels are passive transport systems which move molecules greater than 1,000 times as fast as pumps and carriers. So channels can move things much more quickly. Channels respond to environmental conditions or environmental changes, and these changes can be physical and chemical in nature. The ion channels will allow for rapid movement across the membrane down the concentration gradient. We can talk about action potentials in terms of discussing the movement of these things. And the action potential, if we're looking at action potentials to discuss movement, the action potential is a nerve impulse. And this nerve impulse is going to be generated when the membrane potential is depolarized. And we can use this in the channels. So we will look at or see different types of channels. We'll look at voltage gated. And we'll look at ligand gated or ligand gated. These are 2 different types of channels. When we're talking about these voltage gated channels, we'll look at the potassium and the sodium channels, not the sodium potassium pump, but just the channels that move sodium and potassium ions across. So these voltage gated sodium allow passage of sodium ions. Voltage gated potassium allow passage of potassium ions. In general, when we're looking at these substances, the resting potential for the overall channel will be about 70 millivolts or negative 70 millivolts. And at that point, the channel would be closed. And then when we have changes in the voltage, the channel will open and ions will flow in. So if you're at resting potential and you apply a stimulus that exceeds some threshold value, let's say that threshold value is around negative 45 millivolts, that will result in the voltage gated channel opening and ions flowing through. And that's going to be this depolarization period. So your channel is open and ions can flow through. In the depolarization period, the ions move down their electrochemical gradient. That's going to result in the charge inside the cell changing. Because we're moving positive charges in, we're changing the charge of the overall membrane. When the cell is going to reach a particular voltage, let's say positive 40, because we're changing that charge, that channel will close and inactivate. Okay, so it's open because of a stimulus. Ions are going to flow in. The ions are positively charged. They're flowing in. They're changing that charge of the membrane. At some position, again in this figure, positive 40, that channel closes and inactivates. When that happens, so if we're looking at, say this was the sodium channel, when that happens, the sodium channel reaches a particular point, it's closed. In response to that, the potassium channel would then open up. And the potassium channel would maybe flow in the opposite direction and charges would move in the other way. And that would allow for repolarization because we're moving positive charges now in the other direction. It's possible that we move enough that we have this period where we have hyperpolarization, and that's where we've dropped below what our original resting potential of the cell was. If that happens, in order to get back to this resting potential, we have some of those pumps that will move the substances across to get back to that potential. Now, just note that when we have these action potentials or we have these stimuli that cause these channels to open or close, it's an all or nothing process. They're open or they're closed. There's nothing in between. There are a couple of different ways that we can measure what's going on with a channel and how we've understood how some of these channels function. One is patch clamp conductance measurements, and that's shown here. So this just reveals activities of single channels, and the way that we get this is you can take a pipette and it's placed against a portion of the membrane. You apply suction and that forms a tight seal, and that allows us to measure the activity of that portion of the membrane that is pinched there in that pipette. So it's used to measure activity of a single channel. Now you can have that suction can actually burst the actual membrane, or you can have that excised patch mode where it's kind of turned inside out. But in general, this is a method that's used to allow us to explore what's going on. So this just shows some data that's associated with that patch clamp. Looking at B here, this is going to give an indication of length of time that a channel is in an open state. So downward spikes here will indicate transitions between open and closed state. And we can see when the channel is open and how long it's open for. So we get this information where we're able to see responses in this channel opening and closing. This tetrodotoxin, this is a neurotoxin that's found in pufferfish. What this does is it will actually bind channels tightly, and it's been used in experiments so that we can purify sodium channels so that we can study them. So it's allowed us to study the structure of sodium channels. The sodium channel itself has 4 repeat regions, 5 hydrophobic segments, 1 hydrophilic segment. The potassium channel has 4 repeats, which is similar to sodium. And instead of a neurotoxin, we would use something called a shaker to study that potassium system. So this just shows the overlap in terms of the sodium channel and the calcium channel, and what's involved in terms of just general sequencing of these things. So they're very similar in terms of their structure. Here we see the structure of potassium channel. We see that it has the 4 subunits, the homologous subunits, and then we see the pore size in the center here. There's our pore. We're looking at a top-down view. We will talk here in a second about how that pore size is selective to potassium. So here is another view of our potassium channel. Now, what happens is we have this opening, which is pretty large at the top. So the ions, remember that ions in general, if we're looking at a potassium ion, they are not just free ions floating around in your bodies. Remember that we're mostly water, and ions will tend to be solvated. So each ion has a hydration sphere that's going to be formed around it, and the size of that hydration sphere will depend on the size and the charge of the ion. So these are pretty large, and they will come in, so they need a large space to accommodate this. So the hydrated ion is going to be able to come in to this particular channel. But then our pore is going to start to narrow here. So we see that we have what was pretty wide at the top is narrowing down to about 3 angstroms here. So the potassium has to lose its hydration sphere. Interactions between the ion and the pore have to compensate for the loss of that hydration sphere. So the hydration sphere goes away, and the ions are going to interact with the residues that are within this small portion. Larger ions will be too big to pass through. Smaller ions won't be able to interact with these residues that are in here, and they won't compensate for the loss of the hydration sphere. So in that way, this is like a selectivity filter which is specifically designed for a particular ion, in this case potassium. So this just shows the interaction within that selectivity filter, and how these potassium ions would interact with specific residues that were inside that pore. Again, selectivity is dependent upon size and charge. So here we see different ions, the radius and free energy of hydration that's associated with them. So in order to pass through a membrane, you need to compensate for this hydration free energy. So the interactions that would form between the ion and the pore have to compensate for these. Again, looking at this compensation, if we're looking at potassium versus sodium in the same channel. If this is a potassium channel, we have our substance that here we've got to lose that hydration sphere. And this shows, this downward arrow here, that when the ion interacts with the channel, we're compensating for what we've lost when the hydration sphere comes off. If you take a sodium ion and try to put it in the same channel, here's our sodium ion losing its hydration sphere. That's what it costs to lose its hydration sphere. But when that sodium interacts with the potassium channel, it doesn't quite make up for what we've lost. So the sodium would not pass through this channel. Selectivity for potassium channels, there are 4 binding sites that are present. Potassium will enter one at a time, and charge repulsion will help these ions move through rapidly. So potassium enters the pore one at a time. As it moves through the filter, another potassium will come in. As that comes in, repulsion will occur and it'll kick the ion through, and you'll continue that process of moving ions. So here's our interior potassium comes in, another one approaches. As it comes in, repulsion occurs, which kicks this one through. Another one will come in, repulsion occurs. So these don't like to be near each other. It kicks it through, another one comes in, etc. So you're just moving with that repulsion as we're going across. Sodium and potassium are voltage-gated channels. They change conformation based on potential, based on charge, and that allows for opening and closing. This is just the general structure of this voltage-gated channel. Now we do have voltage sensing paddles that lie in a downward position below the channel. And the voltage channels, you're going to end up with movement of that paddle, which can open or close the membrane. So this is our paddle, which can close. If it moves, it can open the membrane. There are ways to inactivate this channel. One model for inactivation of the channel is the ball and chain model for activation or inactivation. Basically, you have some polypeptide portion, which is the ball, and it's connected to a linker, which is attached to that paddle. When the channel is closed, the ball rotates freely. When the channel is open, the ball is going to find the open site, and it's going to block that pore, so it inactivates the channel. The channel is only open for a brief period, and that allows for membrane inactivation. We also have ligand-gated channels, or ligand-gated channels. Neurotransmitters, like acetylcholine, are responsible for allowing nerve impulses to communicate across synapses. Acetylcholine is released into the synaptic cleft and binds to a channel. And the channel, that's an acetylcholine receptor. So because it's binding, it's a ligand-gated channel. The channel opens and triggers an action potential there. The impulse must be created for the channel to change so that it's open or closed. Now, acetylcholine is the best understood of these types of channels. The binding of acetylcholine will result, again, it's the binding that results in opening and closing. So here we see the application. You have a nerve impulse that's coming through. Synaptic vesicles are going to release acetylcholine. The acetylcholine that's released, it's going to bind postsynaptic membrane, and that changes permeability and triggers an action potential. So the binding of the acetylcholine opens the channel, and the flow is going to continue down through. Here we're just looking at the overall structure of that acetylcholine receptor. There are 4 subunits that form a pentameric ring here. binding of acetylcholine causes a conformation change, which opens the pore. So we can see the pore here. This is just showing open versus closed pore, and you can see that change in size. Open pore is larger, so more can pass through there. Action potential will integrate activity of several ion potentials working together. So the ion channels that we have to be able to communicate with one another. Our channels are not going to function in isolation. They all contribute to an overall potential across the membrane, and that overall potential is controlled by opening and closing different channels at different times. So there is some equilibrium potential that we have to reach. So this figure, the take-home is that there are different ions that have to move, and overall the channels have to cooperate with one another, so we have the appropriate potential across the membrane to reach that equilibrium potential. Initiation of action potential, membrane potential moves from resting towards a sodium equilibrium potential, and then it moves to a potassium equilibrium potential. So these figures are just kind of showing what's going on. We have sodium equilibrium potential, we have potassium equilibrium potential. So if there are different channels that are going to change as the action potential is created. So we need to be able to balance out the equilibrium potentials overall. And we can see that, again, we have ways to activate and inactivate these and control how much of the ions are flowing through. Now, if we do disrupt channel activity, this can lead to a number of different conditions. So if the channels don't work together in a way that balance out overall, conditions like something called long QT syndrome can result. And in long QT syndrome, the recovery of an action potential is going to be delayed. And when we delay the recovery of that potential, you end up with a loss of consciousness, you can also have heart arrhythmia, You can even have sudden death that results. So controlling and regulating these channels is very important. Functioning channels are very important in life. So we've been discussing channels. We've talked about two different types of channels. We've talked about voltage-gated and ligand-gated channels. But there is a third type of channel that we can discuss, and that third type of channel is the gap junction. Now, there are three main functions of gap junctions. The first is intracellular communication. The second is going to be cell nourishment. And the third is embryological development. So three functions of gap junctions. Now, gap junctions will differ from other channels in a number of different ways, and we'll list here 5 different ways that the gap junctions will differ from other types of channels. The first is that we have larger pore sizes with gap junctions. So gap junctions, the pore sizes are about 20 angstroms overall. And remember that in the other channels, we had narrowing of that pore. A second difference is that we can have inorganic and organic substances moving through. So inorganic substances pass. So instead of just ions moving through, or those inorganic portions, we can also have things like glucose moving through. The gap junctions can be open seconds to minutes. Whereas other channels are only going to be open for milliseconds. Gap junctions are going to transverse 2 membranes of cells that are close to one another. So it involves 2 cells rather than just a pore within one cell. And because of that, it's going to connect the cytoplasm of one cell to the cytoplasm of another cell. And because it's two cells that are involved, gap junctions are synthesized by two different cells, by two cells, whereas the other channels are only made by one cell. This is just showing us a picture of gap junctions. Now, a single gap junction will consist of two hexagonal structures that are called hemichannels. And these hemichannels are also going to be referred to as connexons. And these are connected end to end across two membranes. Each of these hemichannels will consist of 6 polypeptide units, and those polypeptide units are going to be referred to as connexins. Each hemichannel will be formed by its own cell, and again, two cells are going to contribute to the overall structure. So here we're seeing more detail on these. You can see this is our space between the two cells. This is the membrane of one cell and the membrane of another cell, and we can see that here. So you see the interior of each cell and the portion that connects the interior of the cells. Each cell has its own portion that contributes to the overall gap junction, and it creates this pore to allow movement between two different cells. Okay, so we've talked about voltage and ligand-gated channels, and we've talked about gap junctions. And all of these are going to move ions and other molecules across membranes. We do have one additional class of molecules, or special class that's going to allow the movement of molecules across, and these are referred to as aquaporins. Okay. Aquaporins are going to be a special class that is going to increase the rate at which water moves across the membrane. So aquaporins allow for specific and rapid movement of water molecules through the membrane. And here we just see the structure of the aquaporin. Now, because we're moving water through that pore, the inside is going to be lined with hydrophilic residues. Hydrophilic so that water can interact with it, and it helps facilitate the movement of water. Okay, so overall, in this chapter, we've looked at pumps and the different types of pumps. So ATPases and ABC transporters. We've looked at carrier proteins and secondary transporters. So antiporters, uniporters, symporters. And we've looked at channels. And with these channels, we've looked at voltage-gated, ligand-gated, and gap junctions. And we've also talked about a special class of transporters called aquaporins that move water. So you want to be familiar with each of these membrane structures, which are active, which are passive transport. Can you describe the process that's going on here? Can you give examples of these things?